In an article published on May 23 in Jama Neurology, researchers describe preliminary research that could open the way to a future test to diagnose ALS. ALS is difficult to diagnose because, currently, there is no single test that can confirm the disease. Doctors are looking for neurological symptoms such as muscle weakness and upper and lower symptoms of motor neurons. They will also do diagnostic tests to exclude other conditions such as cervical spondylosis. A diagnostic test that could confirm SLA would help people get diagnosis earlier and start treatment as soon as possible. Amyotrophic lateral sclerosis is a progressive nervous system disease. It affects the nerve cells of the brain and spinal cord called neurons. The motor neurons control the muscle movement, and the SLA makes them deteriorate and eventually die. The motor neurons lose the ability to send messages to the muscles of the body, affecting voluntary muscle movements. There has been recent progress in the treatment of ALS, but current treatments can only slow down the progression of the disease. This is why it is important to diagnose SLA as soon as possible. It is difficult to diagnose ALS in its early days because there is no known biomarker, said researcher Hirofumi Maruyama, professor at the Graduate School of Biomedical and Health Sciences at the University of Hiroshima, in Japan. The muscle is possible to the biopsy, and the transactive response of the DNA 43 binding protein accumulates in the peripheral nerves inside the muscle. The TDP43 is a protein that plays a key role in motor neurons, and the accumulation of TDP43 can be a biomarker for the early diagnosis of SLA. Previous research in mice has revealed a crucial function of TDP43 in axons, the part of the neuron which sends signals to other neurons. This is important for SLA because axonal degeneration causes weaker motor problems that can be a symptom of SLA. Researchers have hypothesized that the accumulation of TDP43 in the muscular nerve beams could be an early predictor of SLA. To test this theory, the researchers first examined the muscle tissue of 10 people who had confirmed cases of ALS at the time of their and 12 who did not do so. The 10 patients with ALS had TDP43 accumulations in their intramuscular nervous beams, while 12 non-ALS witnesses had no accumulation of TDP43. Then the researchers targeted 114 patients who underwent muscle biopsy and did not have a family history of SLA or another muscular or neuromuscular diagnosis. Among these, 71 had proofs of intramuscular nervous